Welcome all to our channel Sri Rajlakshmi Publication. Agriculture is the backbone of the Indian economy. Indian farmers are cultivating their farms in traditional way, but nowadays farmers are adopting new techniques and others experiences in their farm. The state agriculture universities have a giant share in agricultural development as well as economic and social upliftment of the farmers. The state agriculture universities also play a vital role in nation building. Being a well-wisher of the students and the beneficiaries of these subjects, Sri Rajlakshmi Publication has now decided to publish these subjects in video form on the basis of agricultural development, that is, agronomy, genetics and plant breeding, agri-entomology, horticulture, agricultural economics, agri-extension education, agri-engineering, plant pathology, soil science and agri-chemistry, animal husbandry and dairy science. I express my sincere thanks to all the respected authors of books, research papers, review articles, as well as e resources whose reference has been taken for these subjects and video who have contributed to this publication, and also to the students who refer these books and videos to develop their knowledge updated for their bright future. Suggestions for improvement of this video are welcome. Please comment, like, share and subscribe our channel. That is Sri Rajlakshmi Publication. Sri Rajlakshmi Prakashan published a textbook on Introductory Microbiology. Author Dr. S. A. Raut. Chapter 2nd, History of Microbiology. History of microbiology in brief. Obviously, humans have had to deal with microbes even before the recorded history. The first record of human using comes from ancient tablets from Mid-East. Babylonians were using yeast to make beer over 8,000 years ago and acetic acid bacteria to make vinegar over 6,000 years ago. About 5,000 years ago, Persia, now Iran, region recorded the wine making. The Romans had a god that was specific for microorganisms. The Roman god of mold and mildew was Robigus and Robigo, which means crop rust. Rust is one of the plant disease caused by fungus. God Robigus was very much feared because of crop loss. About 2000 years ago, Romans proposed that diseases were caused by tiny animals, but fundamentalist religions had a strong hold over the progress. The real microbiology history starts from 1600 when people began to make crude lenses and microscopes. Origin of life controversy. Where did microbes come from? Many believed that they arise from simple materials by process of spontaneous generation. This notion had been posited by Aristotle 382 to 322 BC and other Greek philosophers to explain decay and appearance of animals such as flies and frogs, and was widely held as common sense even in 1700 and 1800. Francisco Redi (1626 to 1697) demonstrated that flies did not arise spontaneously from rotting meat by simple experiment. If jar of meat was covered by fine muslin, maggots did not arise. However, the simpler life forms discovered by Leeuwenhoek lacked visible complexity, and most people still believed these could arise spontaneously. John Needham (1731 to 1781), a Scottish clergyman and naturalist, shown that microbes grew in soaps exposed to air, claimed existence of a life force present in inorganic matter that could cause spontaneous generation. One of his more convincing demonstrations was to boil some soap briefly, pour into clean flasks with cork lids, and show that microbes would soon arise. Lazzaro Spallanzani (1729–1799) claimed Needham's organisms came from heat-resistant microbes. If flasks were boiled long enough, 1 to 2H, nothing grew, but Needham countered that prolonged heating destroyed the life force. Louis Pasteur (1822 to 1895) was passionate believer that life only originated from previous life. developed several experiments that finally claims for spontaneous generation. Pasteur filtered air through cotton to trap airborne materials, then dissolved the cotton and examined the particulate matter under a microscope. Many bacteria and spores of other life forms such as molds were present. Since most skeptics kept arguing that overheating killed the life force present in air, Pasteur developed an ingenious experiment using a swan neck flask that allowed fresh air to remain in contact with boiled materials. The long passageway prevented airborne microbes from reaching the nutrient liquid without impeding access to air. One of Pasteur's flasks is still sterile after 100 plus years of being exposed to the air. Pasteur Institute, Paris. Now here you can observe the figure of scientists named Francisco Redi, John Needham, Lazarus Palazzani, Louis Pasteur. Spontaneous generation theory. From earliest times, people had believed in spontaneous generation, that living organisms could develop from non living matter. Even the great Aristotle, 384 to 322 BC, thought some of the simpler invertebrates could arise by spontaneous generation. This view finally was challenged by the Italian physician Francesco Redi, 1626 to 1697, who carried out a series of experiments on decaying meat and its ability to produce maggots spontaneously. Reddy placed meat in three containers. One was uncovered, a second was covered with paper, and the third was covered with fine gills that would exclude flies. Flies laid their eggs on the uncovered meat and maggots developed. The other two pieces of meat did not produce maggots spontaneously. However, flies were attracted to the gauze-covered container and laid their eggs on the gauze. These eggs produced maggots. Thus, the generation of maggots by decaying meat resulted from the presence of fly eggs, and meat did not spontaneously generate maggots as previously believed. Similar experiments by others helped discredit the theory for larger organisms. Leeuwenhoek's discovery of microorganisms renewed the controversy. Some proposed that microorganisms arose by spontaneous generation even though larger organisms did not. They pointed out that boiled extracts of hay or meat would give rise to microorganisms after sitting for a while. In 1748, the English priest John Needham, 1713 to 1781, reported the results of his experiments on spontaneous generation. Needham boiled mutton broth and then tightly stoppered the flask. Eventually, many of the flasks became cloudy and contained microorganisms. He thought organic matter contained a vital force that could confer the properties of life on non-living matter. A few years later, the Italian priest and naturalist Lazzaro Spallanzani, 1729 to 1799, improved on Needham's experimental design by first sealing glass flasks that contain water and seeds. If the sealed flasks were placed in boiling water for three-fourths of an hour, no growth took place as long as the flask remained sealed. He proposed that air carried germs to the culture medium, but also commented that the external air might be required for growth of animals already in the medium. The supporters of spontaneous generation maintain that heating the air in sealed flasks destroyed its ability to support life. Several investigators attempted to counter such arguments. Theodore Swan, 1810 to 1882, allowed air to enter a flask containing a sterile nutrient solution after the air had passed through a red hot tube. The flask remained sterile. Subsequently, George Friedrich Schroeder and Theodor von Dusch allowed air to enter a flask of heat-sterilized medium after it had passed through sterile cotton wool. No growth occurred in the medium even though the air had not been heated. Despite these experiments, the French naturalist Félix Pauchet claimed in 1859 to have carried out experiments conclusively proving that microbial growth could occur without air contamination. This claim provoked Louis Pasteur (1822 to 1895) to settle the matter once and for all. Pasteur first filtered air through cotton and found that objects resembling plant spores had been trapped. If a piece of the cotton was placed in sterile medium after air had been filtered through it, microbial growth appeared. Next, he placed nutrient solutions in flasks, heated their necks in a flame, and drew them out into a variety of curves while keeping the ends of the necks open to the atmosphere. Pasteur then boiled the solution for a few minutes and allowed them to cool. No growth took place even though the contents of the flask were exposed to the air. Pasteur pointed out that no growth occurred because dust and germs had been trapped on the walls of the curved necks. If the necks were broken, growth commenced immediately. Pasteur had not only resolved the controversy by 1861 but also had shown how to keep solutions sterile. The English physicist John Tyndall (1820 to 1893) dealt a final blow to spontaneous generation in 1877 by demonstrating that dust did indeed carry germs and that if dust was absent, growth remained sterile even if directly exposed to air. During the course of his studies, Tyndall provided evidence for the existence of exceptionally heat-resistant forms of bacteria. Working independently, the German botanist Ferdinand Kohn (1828 to 1898) discovered the existence of heat-resistant bacterial endospores. Number 1. The spontaneous generation experiment. Pasteur's swan neck flask used in his experiments on the spontaneous generation of microorganisms. Here you can see the figure of Pasteur's apparatus. Name, sterilize, water trap, no growth, mix broth with water, broth spoils. Number 2. Disprove of spontaneous generation theory. At that time, the age-old idea of spontaneous generation theory was the dominant one. The idea that organisms originate directly from non-living matter, life from non-living, also called as abiogenesis, a not biolife genesis origin. Example, maggots were developed spontaneously via recombination of matters in rooting materials. Example, meat. The microbiology starts when the disproof of SG theory. Louis Pasteur (1822 to 1895) and disproval of spontaneous generation theory. He performed Gosenick experiment. The nutrient of flask was heated and the untreated, unfiltered air could pass in or out, but the germs settled in the Gosenick and no microbes were observed in the nutrient solution. His concept of germs theory of disease means germs are responsible for the disease, not the inert matter, hence the SG theory. Contributions of Louis Pasteur (1822 to 1895) disproved the SG theory, discovered that fermenting fruit to alcohol by microbes. From now the fermentation started, sorted different microbes giving different taste of wine. He selected a particular strain, yeast, for high quality wine. He developed a method to remove the undesired microbes from juice without affecting its quality. Heating the juice at 62.8 degrees Celsius for half an hour did the job. This technique is called as pasteurization, which is commonly used in the field of milk industry. He discovered that parasites, protozoa causing pebrine disease of silkworm. He suggested that disease-free caterpillars can eliminate the disease. He isolated the anthrax-causing bacilli from the bloods of cattle, sheep, and human beings. He also demonstrated the virulence ability of microbe to cause disease of bacteria. He developed vaccine, a killed or attenuated microbe to induce the immunity against rabies from the brains and spinal cord of rabbit. John Tyndall (1820 to 1893) proved that dust carries the germs, and if no dust in the air, the sterile broth remain free of microbial growth for indefinite period. He also developed a sterilization method, tyndallization, referred as intermittent or fractional sterilization. The subsequent cooling and heating by steam for 3 days will remove the germs and their spores. Martinus William Bisering (1851 to 1931) developed the enrichment technique to isolate various group of bacteria. Isolated sulfur reducing bacteria and sulfur oxidizing bacteria from soil. Isolated free living nitrogen fixing bacterium Azotobacter from soil. Root nodulating bacterium Rhizobium lactobacillus green algae were identified by him. He confirmed the tobacco mosaic virus causes disease and it incorporated in the host plant to reproduce. Sergei Vinogradsky, 1856 to 1953. The following are the contributions of Vinogradsky to soil microbiology. Microorganisms involved in N cycle, C cycle, S cycle, nitrification process in soil, autotrophic nutrition of bacteria, chemolithotropic nutrition of soil bacteria, discovered anaerobic nitrogen fixing bacterium Clostridium pasturanium. Walter Hesse and Fanny E. Hesse, 1883. They use agar instead of gelatin for preparation of media. Agar goes to solution at 100 degrees Celsius and solidifies at 45 degrees Celsius. Till now, this was not replaced by any other substance. Here you can see the figure of scientist. Joseph Lister, 1878. Develop pure culture technique. Pure culture referred as the growth of mass of cells of same species in a vessel. He developed the pure cultures of bacteria using serial dilution technique. He also discovered that carbolic acid to disinfect the surgical equipments and dressings leads the reduction of post-operational deaths infections. Alexander Fleming, 1928, identified Penicillium notatum inhibiting Staphylococcus aureus. 1929, discovered antibiotic penicillin, important milestone in medical microbiology, found that natural substances having antimicrobial activity, saliva, nasal mucus, work on Staphylococcus aureus inhibition of growth due to penicillin. Florey and Chen isolated penicillin in pure culture. Selman A. Waxman, 1945, identified streptomycin antibiotic from soil bacterium. He also coined the term antibiotics, referring a chemical substance of microbial origin which is in small quantity exert antimicrobial activity. 1927, wrote the book on principles of soil microbiology. In 1939, Waxman and his colleagues undertook a systematic effort to identify soil organisms producing soluble substances that might be useful in the control of infectious diseases, what are now known as antibiotics. Within a decade, 10 antibiotics were isolated and characterized, three of them with important clinical applications: actinomycin in 1940, streptomycin in 1944, and neomycin in 1949. 18 antibiotics were discovered under his general direction. Germ theory of disease. Bacteria are found everywhere in water, soil, and even air. These small prokaryotic cells, typically from 0.2 to 1 um in length, are capable of living in boiling water, frozen ground, acid volcanoes, and at the bottom of the ocean. They can reproduce by doubling with a generation time of 20 minutes or survive for centuries in a resting stage. In natural waters, lakes, streams, oceans, their generation time is around 1 day. In soils, they live in a film of water around plant roots or other particles, and their activity is dependent on the temperature and the amount of available moisture. In general, bacteria are found in concentrations of 10 raised to 6 cells by ml of water in surface waters and 10 raised to 9 cells by ml of soil in soils and sediments. Robert Koch, 1843 to 1910, the father of microbial techniques. Robert Koch, a German physician, is well known to the world of microbiology for his significant contributions, especially in the area of microbial techniques. He introduced aniline dyes for staining bacteria, used agar-agar and gelatin to prepare solid culture media, stressed the need for pure culture to study microbes in details, confined germ theory of disease, and laid down postulates to test the pathogenicity of causative agents. He also discovered the causal organisms of anthrax disease of cattle, Bacillus anthracis, and tuberculosis, Mycobacterium tuberculosis. Robert Koch was particularly concerned with this problem and at first, he cultured bacteria on solid fruits and vegetables such as slices of boiled potato, but many bacteria did not grow on such substrates. Then he perceived that it would be far better if a well tried liquid medium could be solidified with some clear substance. Coach 1881 tried gelatin as a solidifying agent and succeeded in developing solid culture media, but gelatin, the first solidifying agent used, had serious disadvantage of becoming liquid above 28 to 30 degrees Celsius, which is below the optimum temperature for the growth of human disease producing bacteria. However, Koch replaced gelatin by agar in 1883 to 84 on the recommendation of F.E. Hesse, a German housewife who had gained experience with the characteristics of agar in the process of making jelly. Agar is still frequently used as solidifying agent in microbiological laboratories. The development of solid culture media to grow pure culture was of fundamental importance and may be considered one of the Koch's greatest contributions. Besides developing solid culture media using gelatin and agar, Koch also evolved methods to place microbes on glass slides and color them with aniline dye stains so that the individual cells could be seen more clearly under the microscope. Koch postulates. These postulates were developed for identifying the organism associated with a particular disease by Robert Koch. Number 1. The microorganism must be present in every case of the disease but absent from healthy organisms. Number 2. The suspected microorganism must be isolated and grown in a pure culture. Number 3. The same disease must result when the isolated microorganism is inoculated into a healthy host. Number 4. The same microorganism must be isolated again from the disease host. Initial attempts to isolate microbes use sliced potatoes or nutrient media containing gelatin, not ideal media. Then Fanny Hesse, wife of lab worker, suggested agar, a gelling agent used in cooking. Agar rapidly became the standard gelling agent for microbial isolation because it is relatively inert. Only some marine microbes have enzymes to digest agar. Agar only melts at high temperatures, 100 degrees Celsius. Once melted, it remains liquid until about 45 degrees Celsius, at which point it gels. Koch's success at identifying anthrax with bacterium Bacillus anthracis led both Koch and Pasteur to identify the causes of many diseases, cholera, tuberculosis, plague, etc. Over the next few decades, late 1880s, the golden age of microbiology, 1870 to 1920. Note that many microbiologists would regard the present as a new golden age since the development of molecular biological techniques, PCR, molecular phylogeny, and other developments have revealed many new insights and open a world of new research directions and ways of understanding microbes. History and development of microscope. Timeline of the microscope. 14th century. Spectacles first made in Italy. 1590. Two Dutch spectacle makers and father and son team, Hans and Zacharias Jensen, created the first microscope. 1667. Robert Hooke's famous Micrographia is published, which outlines Hooke's various studies using the microscope. 1675. Anton van Leeuwenhoek, who used a microscope with one lens to observe insects and other specimens. Leeuwenhoek was the first to observe bacteria. 18th century, as technology improved, microscopy became more popular among scientists. Part of this was due to the discovery that combining two types of glass reduced the chromatic effect. 1830, Joseph Jackson Lister discovers that using weak lenses together at various distances provided clear magnification. 1878, mathematical theory linking a resolution to light wavelength is invented by Ernst Abbe. 1903, Richard Sigmondi invents the ultramicroscope, which allows for observation of specimens below the wavelength of light. 1932, transparent biological materials are studied for the first time using Fritz Zernick's invention of the phase contrast microscope. 1938, just 6 years after the invention of the phase contrast microscope, comes the electron microscope developed by Ernst Ruska, who realized that using electrons in microscopy enhanced resolution. 1981, 3D specimen images possible with the invention of the scanning tunneling microscope by Gerd Binning and Henrik Rohrer. Origin. The origin of the word microscope according to the online etymology dictionary is as follows. 1656, from Mood L. Microscopium lit, an instrument for viewing what is small, from GK Micro QV plus Scorpion means of viewing from scorpion look at microscopic of minute size is attested from 1760s history of the compound microscope. Just as the Greeks had a fully functioning radiant heating system operating 2000 years before those only now being introduced in the US, so the origins of the compound light microscope appear to be traced not to Holland, England, or France, but to China, which is perhaps appropriate given the present predominance of China in supplying compound light microscopes. The water microscope. According to an ancient Chinese text, the Chinese viewed magnified specimens through a lens at the end of a tube, which tube was filled with varying levels of water according to the degree of magnification they wished to achieve. Ingenious, effective, and repeatable in the home today. That this occurred some 4000 years ago in the Chao Fu dynasty and more than 3500 years before the father of modern microscopy was born is quite remarkable. That these Chinese ancients achieved magnification levels of 150 times today's standard or 100 move is breathtaking. It is as if they developed a town car that achieved Mach 2. If they did build such a car, no reference to it has ever been found. Similarly, there is no further known reference to such a compound microscope device until we come back to the Greeks again. No less a person than Aristotle describes the working of a microscope in some detail. The Greeks certainly made good use of curved lenses, which are an essential component of any stereo or compound microscope. Ancient Greek boys probably shared every American boy's sense of triumph of using a curved lens or magnifying glass to start a fire. The Greeks, however, also used it for surgical procedures, not on ants as little boys are wont to do, but on people to cauterize wounds and lesions caused by leprosy and so forth. Ancient Egyptians and Romans also used various curved lenses, although no reference to a compound microscope has been found. The Greeks did, however, give us the word microscope. It comes from two Greek words, Ippos, small, and occultive view. However, while ancient Chinese, Greeks, and Romans all applied their infinite wisdom to the issue, there is no known reference to either the use of artificial light or to multiple lenses. In other words, we can give great credit to the ancients for their foresight and achievements, but we have to look elsewhere to uncover both the first light and compound microscope. Incredibly, the next historical references with anything at all to do with microscopes, or more accurately, optics, is 1200 years after Rome was sacked, and even then, the references are only to the use of lenses in the invention of spectacles. Put another way round, some of the smartest people the planet has ever produced played and worked with single lenses for several thousand years without taking it further. Spectacles. Then, within just a few short years in Tuscany, Italy, two men claimed to have independently invented spectacles. The evidence their tombstones, one Salvano di Armento degli Amati died in 1284 in Florence and claimed to have kept the process secret. The other, Alessandro della Spina, died in 1317 and claimed to have revealed his process. In any event, a local monk, Girodina da Rivalta, gave a sermon in 1306 in which he enthusiastically endorsed spectacles as a terrific invention and in passing indicated that they had been in use for about 20 years. Finally, in 1289, another local from the Popozo family bemoaned that I am so debilitated by age that without the glasses known as spectacles, I would no longer be able to read or write. Telescopes. At about the same time, it appears that lenses were being used in early telescopes. In the 13th century, the Englishman Roger Bacon discusses them at length. Both spectacles and microscopes are relevant to microscopes because they trace the increasingly sophisticated use of lenses, the essential optical component of any microscope. Then, a mere 200 to 300 years later, we find a plethora of references and hard evidence of both telescopes and microscopes. The Renaissance had arrived, and with it, an abundant flowering in the arts and sciences. Most importantly, with the invention of the printing Horus, ideas and developments could be spread easily and rapidly. As a result, Thomas Digges' work on the telescope in England in the mid 16th century and Hans Lippershey's work, which included applying for a telescope patent, were transmitted to others, including no less a genius than Galileo. Galileo immediately began to work with lenses. In a short time frame, he developed an improved telescope with a focusing device and went on to conquer the stars. That said, we should also pay tribute to Sir Isaac Newton, who around the same time in the UK invented the reflecting telescope. But what of microscopes? Well, the same Hans Lippershey and his son Zacharias Hansen was experimenting with a variety of lenses. In the late 1590, they used several lenses in a tube and were amazed to see that the object at the end of the tube was magnified significantly beyond the capability of a magnifying glass. They had just invented the compound microscope. That is to say, that they had discovered that an image magnified by a single lens can be further magnified by a second or more lenses. Then, in the mid 17th century, an Englishman, Robert Hooke, and a Dutchman, Anthony van Leeuwenhoek, took the microscope to new levels. Hooke was a sickly genius who loved to experiment. He did so across a huge range of scientific fields of study and with prolific success. He invented the universal joint, the iris diaphragm, another key component of many modern light microscopes, a respirator, an anchor, escapement, and balance spring for clocks. Robert Hooke. He also worked out the correct theory of combustion, devised an equation describing elasticity that is still used today, Hooke's law, and invented or improved meteorological instruments such as the barometer, anemometer, and hygrometer, and so on. Most of all, however, he is known for micrographia, his studies with a microscope published in 1665. Micrographia became an overnight sensation not just for what he described but for the superb drawings that he made. He described a new world alongside exquisite drawings of the stinging hairs on a nettle, a flea, and most famously of all, the honeycomb structure or cells of a cork. It was Hooke who coined the term cells when describing living tissue. Interestingly, while Hooke did use a compound microscope, he found that it much strained and weakened his sight. For his micrographia, he preferred to use a simple single lens microscope made of gold and leather and illuminated by a candle, perhaps the first light microscope. Antony van Leeuwenhoek, the father of the microscope. It was Leeuwenhoek, however, who lived at the same time as Hooke and drew on Hooke's work to take microscope design to new levels of sophistication. As a draper, he used a simple microscope to examine cloth. As a scientist, he began to experiment with new ways of grinding lenses in order to improve the optical quality. In total, he ground some 550 lenses, some of which had a linear magnifying power of 500 and a resolving power of 1 millionth of an inch, an astounding achievement. Leeuwenhoek detailed these achievements in almost 200 letters to the Royal Society in London, where no less a person than Robert Hooke validated them. The result of all this work was a simple single lens handheld microscope. The specimen was mounted on the top of the pointer, above which lay a convex lens attached to a metal holder. The specimen was then viewed through a hole on the other side of the microscope and was focused using a screw. Perhaps his most famous experiment came in 1674 when he viewed some lake water. I now saw very plainly that these were little eels or worms lying all huddled up together and wriggling just as if you saw with the naked eye a whole too full of little eels and water with the eels swimming among one another and the whole water seemed to be alive with these multifarious animalcules. This was for me among all the marvels that I have discovered in nature the most marvelous of all, and I must say for my part that no more pleasant sight has ever yet come before my eyes that these many thousands of living creatures seem all alive in a little drop of water moving among one another, each several creature having its own proper motion. He had discovered bacteria. He had earned his title of the father of the microscope. Interestingly, it took until 1839, nearly 200 years later, before cells were finally acknowledged as the basic unit of life. 18th, 19th centuries. The next major step in the history of the microscope occurred another 100 years later with the invention of the achromatic lens by Charles Hall in the 1730s. He discovered that by using a second lens of different shape and refracting properties, he could realign colors with a minimal impact on the magnification of the first lens. Then in 1830, Joseph Lister solved the problem of spherical aberration. Light bends at different angles depending on where it hits the lens. By placing lenses at precise distances from each other, combined these two discoveries contributed towards a marked improvement in the quality of image. Previously, due to the poor quality of glass and imperfect lens, microscopists had been viewing nothing but distorted images, somewhat like the first radios were extremely crackly. It is worth remembering that up until now, each new stride has been in the quality or application of the lenses. Then in 1863, one of the several new manufacturers of microscopes, the Inst Litz company, addressed a mechanical issue with the introduction of the first revolving turret with no less than five objectives. This improvement was quickly followed in 1866 when Carl Zeiss recruited East Abbey as his director of research at the Zeiss Optical Works. Abbey laid out the framework of what would become the modern computational optics development approach. He made clear the difference between magnification and resolution and criticized the practice of using eyepieces with too high a magnification as empty magnification. By 1869, his work produced a new patented illumination device, the Abbe condenser. Abbe condenser. Abbe's work on a wave theory of microscopic imaging, the Abbe condition, made possible the development of a new range of 17 microscope objectives. Three of these were the first immersion objectives, and all were designed based on mathematical modeling. As Abbe noted, his creations were based on a precise study of the material used. The design concerns are specified by computation to the last detail. Every curvature, every thickness, every aperture of a lens, so that any trial and error approach is excluded. From here on, microscopes were designed based on sound laws of physics rather than the trial and error that had characterized the pioneers. At the same time, a number of companies set up specialized manufacturing plants focused on manufacturing precision microscopes. Research and development continued to bear fruit. In 1880, the first microtomes began to be used that enable significantly thinner samples to be prepared in order to improve sample. In 1893, another Zeiss employee August Kohler figured out an unparalleled illumination system that is still known as Kohler illumination. Using double diaphragms, the system provides triple benefits of a uniformly illuminated specimen, a bright image, and minimal glare. In other words, Kohler achieved an almost perfect image. The mass market for microscopes had arrived at the same time as precision engineering, and it is little wonder that a plethora of stunning results were obtained. In 1879, Walter Fleming discovered cell mitosis and chromosomes, an achievement recognized as one of the 100 most important scientific achievements of all time. 20th century. At the turn of the 19th 20th century, Louis Pasteur invented pasteurization while Robert Koch discovered his famous or infamous postulates, the anthrax bacillus, the tuberculosis bacillus, and the cholera vibrio, UV and phase. By 1900, the theoretic limit of resolution for visible light microscopes, 2000 angstroms, had been reached. In 1904, Zeiss overcame this limitation with the introduction the first commercial UV microscope with resolution twice that of a visible light microscope. In 1930, Fritz Zernick discovered he could view unstained cells using the phase angle of rays. Spun by Zeiss, his phase contrast innovation was not introduced until 1941, although he went on to win a Nobel Prize for his work in 1953. You can see the image of phase and electron microscope. Electron microscopes. In 1931, Max Knoll and Ernst Ruska invented the first electron microscope that blasted past the optical limitations of the light. Physics dictates that light microscopes are limited by the physics of light to 500x or 1000x magnification and a resolution of zero point 0.2 micrometers. Knoll and Ruska built a transmission electron microscope (TEM), one that transmits a beam of electrons as opposed to light through the specimen. The subsequent interaction of the beam of electrons with the specimen is recorded and transformed into an image. Then in 1942, Ruska improved on the theme by built the first scanning electron microscope (SEM) that transmits a beam of electrons across the specimen. Ruska's principles still form the basis of modern electron microscopes that can achieve magnification levels of up to 2 million times. The second major development for microscopes in the 20th century was the evolution of the mass market, started in the 19th century when Leeds claimed to have exported 50,000 microscopes to the US. This trend accelerated in the 20th century. As a result, a large number of manufacturers sprang up to offer more competitively priced alternatives to established European companies such as Zeiss and Leeds. Digital microscopes. Digital microscopes allow for live image transmission to a TV or computer screen and have helped revolutionize microphotography. Digital microscopes simply integrate a digital microscope camera on the trinocular port of a standard microscope. An alternative and more flexible solution is simply to place a digital microscope camera on a trinocular microscope. Dinolite, one of the more original innovations in the 21st century, has been Dinolite digital microscopes. Dinolite are handheld digital microscopes not much larger than a fat pen. They offer low power zoom capability with magnification up to 500x. They have had a marked impact on industrial inspection applications. For more videos and updates, please don't forget to subscribe our channel, that is Sri Raj Lakshmi Publication, as your more subscription encourages us to upload more videos. So please don't forget to subscribe. Thank you.